Hi everybody, thanks for joining today. This is Jason Key at SBGrid in Boston, Massachusetts. This is our weekly webinar series. We have our entire lineup posted on our webpage at sbgrid.org. It is my pleasure to welcome Steve Meisburger from Cornell. So Steve, thank you for agreeing to give a talk today. I have to say, I'm very excited to see X-ray in your title because I feel like I don't get to see that enough these days. And Steve's going to tell us about deconvolving protein dynamics in solution and crystals with X-ray scattering. Go right ahead.
All right, so let me see if I can share my screen.
While he's getting set up, feel free to send questions during the talk in the chat. We'll sort of gather those up and save them for the end. There'll be time for discussion to unmute, and you'll be able to discuss with Steve directly if you'd like, or we can pass your question on. So with that, whenever you're ready.
Great. Thank you, Jason, for that introduction. And thank you as well to the organizers of this seminar for inviting me and for hosting this great weekly seminar series. So the title of my talk, Deconvolving Protein Dynamics in Solution and Crystals with X-ray Scattering. So all those words will make more sense once we get going. So before I start, I just want to introduce some of the people who did the work. So this is the Ando lab at Cornell, where I'm a research associate. And I've just put some names on here. These are names of my co-authors on some of the papers I'll show today, some of the work. So one of them is Darren Shu, who's a graduate student, as well as Will Thomas. And then this is Nozomi Ando. Now, some of the work was also begun when the lab was at Princeton. So this is actually a photo from the Princeton group. And in this picture, you'll see David Case, who's a professor at Rutgers. And he did a sabbatical with us, actually, for a year. And so some of the work that I'll talk about in the second half was a result of that very close collaboration. Okay, so as structural biologists, we all know that protein structure determines function. So proteins fold into three-dimensional shapes. And we actually, these days, know a whole lot about protein structure. So the PDB has 176,247 entries as of a couple of days ago. That's a lot of information about protein structure. Here's a particular entry in the PDB. This is an enzyme that I like a lot. This is the coordinate iron sulfur protein methyltransferase complex. And okay, so we basically know where all of the atoms in this protein are. So the other piece of this picture is function. So do we understand how proteins work? Okay, so let's look at this methyltransferase enzyme. Here it has a couple of cofactors. Here's a vitamin B12. Here is folate. And so the function of this protein, as I said, is a methyltransferase. So it's going to move a methyl group from folate to the cobalt atom on B12. Now, how does this happen? Those two atoms are very far apart. So the methyl group isn't just going to fly through space. The protein must change its conformation in order for the reaction to occur. So based on this structure, do we understand how those protein motions happen? The answer to that, unfortunately, is largely no. So just because we have the structure doesn't mean we understand how the protein moves. Why not? And this is, I think it's because, at least today, looking at a protein structure, the design principles for proteins are not really self-evident. And I'll make this a little more clear in a different example. Okay, so as a biophysicist, like myself, this is kind of what we hope. We'd hope that a protein structure is like a schematic. So a protein being a complicated circuit, once we have the schematic for this complicated circuit, in this analogy, we would understand everything about it. But that turns out not to be true. It's more like this XKCD cartoon, right, where you have just complication everywhere, and you don't know where to focus your attention. So you might spend your whole career studying this resistor network, for example, but really this is just an incidental feature of the protein structure, and it's some other piece that's really important. So this is kind of the reality. What's missing is a dynamic picture. And I'm using the word dynamics very loosely here, so it's meaning how the structure evolves in time and or the ensembles of structures in equilibrium, or the fluctuations in the structures, or the interactions between different proteins or proteins with ligands. Basically everything we'd want to know that we can't get from a single static picture. Now, in the case of this methyltransferase, actually there are multiple crystal structures that trapped it in different conformational states. And this video is showing a morph between some of those crystal structures. What you can see is that indeed, the protein is designed in such a way that the B12 binding domain is able to pivot and move this cobalt atom closer to the folate. Okay, so we need this dynamic picture to really understand protein function. Now, there's another aspect of crystal structures that I want to get into detail about, and that is crystallographic B factors. So crystallographers will know that when you refine a protein structure at high resolution, you refine, in addition to the coordinates of every atom, X, Y, and Z, you also refine this B factor. The B factor is proportional to the mean squared displacement of the atom from its average position. So this has the potential to have some information about dynamics. And indeed, if we color the methyltransferase structure by B factor, what you can see right away is that the B12 binding domain, which was mobile, lights up in red. And so you say, oh, this actually kind of agrees with what we think is happening here. But what do these B factors really represent? Is it the case that this B12 binding domain is moving as a rigid body in the crystal? Or are the atoms in this domain simply more disordered? So we can't tell from the B factors alone. We need new tools. And so I think if we want sort of ideal set of tools for this dynamic structural biology, there are sort of three requirements. First, we need a technique that's sensitive to structural fluctuations. Second, we need some way to isolate the signals that are important for biological function, because maybe not all fluctuations are going to be important. We also need some way to interpret the data with sort of appropriate models. And in the Ando lab, I'll tell you about the approach that we're taking. So the technique we love for this is X-ray scattering. I'll say more about X-ray scattering in a minute. To isolate signals that of interest to biological function, we're going to need some algorithms. And in particular, X-ray scattering signals, as you'll see, are always mixtures of many different components. And so the sorts of algorithms we're going to use are those that unmix mixed signals. And so I've chosen to call these deconvolution. And then models, we're going to bring in physics and chemistry. Okay, so this is a talk for SBGrid. So all of you are interested in software. And so I'm actually going to spend a little more time talking about algorithms than I maybe normally would in a talk like this. So this will be fun. The first part is going to focus on small angle scattering. So scattering from proteins in solution. The second half will be about diffuse scattering. So this is scattering from crystals. All right, so first, why X-ray scattering? So if you shine X-rays on a dilute solution of proteins, you can observe a scattering pattern. And this is sort of the ultimate disordered system. And yet you can still get information about protein shape. So this is sort of one of the techniques we use a lot. Now, many of you know, of course, that if you shine X-rays on a crystal of proteins, you can observe a diffraction pattern that tells you about the average structure of that protein at high resolution. Now, what happens if there's some disorder in your crystal? So I've kind of drawn this as maybe packing sort of defects in the crystal where each protein is in a different position slightly, but you could also imagine each protein having a slightly different conformation. If you shine X-rays on that sort of a crystal, what you see is a superposition of two patterns. First, you have the intense Bragg peaks that you see in normal diffraction, and in the background, you see a cloudy diffuse scattering pattern. And so that will be the sort of subject of the second part of the talk. So first, I'll talk about small angle scattering and the techniques we've developed to analyze that sort of data. So as I mentioned, SAXS is really ideal for dynamic studies. What we do is we take this solution scattering pattern and we integrate at values of constant Q. Q here is the momentum transfer, and it's proportional to the scattering angle roughly. And when you do that, you get a one-dimensional scattering profile as shown on the right. So intensity versus Q. And the great thing about SAXS is that because you're in solution, you can be close to the native state of the protein. You can observe protein-protein interactions. You can very easily alter the environment of the protein and observe how structure changes. So you can do things like ligand titrations, time-resolved experiments. There's one major limitation of SAXS, though, and that is that it's low information. So shown on the right, so the top here is the SAXS profile. This profile is related by a Fourier transform to the pair distance distribution function. This is, you can think of as a histogram of all the electron-electron distances in the protein. And this histogram goes to 0 at the maximum dimension of your particle, of your protein. So we call that D max. And because of this Fourier transform relationship, Nyquist-Shannon theorem tells us that we can represent all of the information in this P of R function by a set of discrete samples of intensity, and the spacing of those samples is pi over D max. And that implies immediately that the number of sort of samples that we're effectively getting in our scattering profile is like Q max times D max over pi. And for SAXS data, that's typically 10 to 20 unique pieces of information. Now, what's amazing about SAXS is often this is enough to reconstruct a unique shape at low resolution. But we're studying mixtures, and in that case, we're trying to learn the structure of many different things in solution, and so this poses a challenge. So how can we increase the information content? One way is just to collect a lot of scattering data. So, and actually there's a bunch of techniques that automatically generate these large data sets. One of them is SAXS with inline chromatography. So here we're just flowing protein over a column, and then it elutes and goes through the X-ray beam. So we end up with this data set of I versus Q versus time, where we have peaks. You can also do ligand titrations, as I mentioned, and of course, time resolved measurements. But once we have these big data sets, we need some way to analyze what's happening in these mixtures. So, all right, so to understand this from a mathematical perspective, the thing to appreciate is that SAXS intensities are just additive in dilute solution. So in this analogy, our proteins are fish, and we have yellow fish and green sharks. And if the yellow fish and the green sharks don't interact with each other, then the scattering from this mixture is just the sum of the scattering we would have gotten with a pure solution of yellowfish and a pure solution of green sharks. And so that's actually pretty simple to describe mathematically. All right, so here's kind of what our model is. So we have intensity versus Q, and maybe time or some other variable is just a sum over the product of component scattering profiles and see how their concentrations evolve in time. All right, so given a bunch of measurements of I versus Q, the challenge now is to deconvolve the individual components. So to figure out what these Y versus Q functions and C versus T functions actually are. Okay, so this is made a little easier when you think about it in terms of linear algebra. So you can simply discretize these functions, which is natural because you measure at discrete values of Q and discrete values of time, for example. So then this thing just turns into a matrix product equation. So the intensity, which is now a two-dimensional matrix with, rows are Q and columns are time. This is now just the product of two other matrices, Y and C, where Y and C have just the component scattering curves as their columns. C has the concentration profiles as columns. Okay, so we can use matrix factorization methods to try and solve this problem. Now, one method that is quite popular to use is singular value decomposition. This has sort of been used for SAXS data since the mid-90s, and it's very powerful technique. So you can take your data in a matrix here, which has your intensities in the columns, and just automatically decompose it into the product of three other matrices. So you have matrices U, S, and V. S is a diagonal matrix, and along the diagonal you have positive numbers in decreasing order, which are the singular values. And these tell you sort of how important each component is in the final data set. And then U and V are the components. So U, these are the components that resemble SAXS profiles, and V are the components that resemble concentrations. So this is great. So it gives you a way of decomposing any data set you have into the product of individual components and then also their weights. Okay, but there's a limitation, and that is that these basis vectors that we get from SVD are usually non-physical. And this is an example to kind of show you what they look like. Here we did some anion exchange chromatography coupled SAXS. This is what the data set looks like. There are a couple of peaks here. And when we just run SVD, we get, okay, 4 components. What are those components? These are the concentrations that we get, and these are the intensities. And you can immediately see that this is not all that useful. The elution peaks didn't separate, so we would kind of suspect that maybe one of these peaks is one species and the other peak is a different species. But in this scheme, there's sort of everything has 2 peaks. Also, these intensities are negative, which can't be right, and they oscillate around 0, which can't be right. So SAXS intensities from proteins are always positive. Okay, so what do? It turns out that other deconvolution methods exist that can sort of give you physical basis vectors, but they're not very general. So one of them is you can do model fitting. So if you have a model for the kinetics and the time constant, the rate constants, you can do a fit. If you know the peak shape in your chromatogram, you can fit that. You can also apply soft restraints, things like sparsity or positivity restraints on the basis vectors. Or you can do another technique that we've used actually quite a bit in the lab, which is evolving factor analysis. And this works really well. I'm not going to say anything more about it, but you can read about it in a couple papers that are at the bottom here. So the problem is, model fitting, you have to know the model in advance, and oftentimes you don't really know what your kinetic scheme is, for example, until you measure it. Soft restraints turn out to be rarely sufficient for small angle scattering to give you a unique solution. And then this evolving factor analysis, as I said, works well, but only for SEC SAXS. And we want to analyze sort of a broad range of data sets. Okay, so how do we incorporate prior information in a more flexible way? Okay, we developed this method that we call REGALS. And the idea is you have your data set, you run it through the method, and you get physical basis vectors out, concentration profiles and SAXS curves. The way that we do that is we express the basis vectors using parametric curves that embody sort of what we mean by physically meaningful solutions. Okay, so what do I mean by that? We use restraints such as smoothness. So you might say that concentration profiles have to be smooth or SAXS profiles have to be smooth. We also use a compact support. Now this means, for example, in chromatography, that a peak has to have a sort of finite range, or that in real space, the P of R function has to have a finite range. In other words, it has to go to 0 at some value d max. And then the user will build a model by mixing and matching different curve types to describe their experiment. And this is where you would need to bring in a little bit of prior information. And then this whole model is refined globally. Okay. So here are a few different basis vector types that we've implemented. So on the left, this is showing the two options for concentration. So you could either have a simple basis vector type. That just means that every value of time or position here, x, that you measure is gets its own parameter. Or you could have a smooth basis vector. This would be, for example, for a chromatography peak, you would have a set of control points, and you can linear interpolate between those control points. And then this curve also would be forced to go to 0 at two values, x min and x max. So this will force the peak to only have a finite support. And then there's something similar in I versus Q space. So we have simple and smooth basis vectors. And then we add one additional one. This is the real space basis vector. The way that works is you have your parameters are defining a P of R function in real space, which goes to 0 at a value of d max. And this is Fourier transformed to give the I versus Q profile. Okay. Now, I need to say something about global optimization of this model because it has some properties that make it tricky. So the model for SAXS deconvolution, as I said, is a product of two matrices, y times c, and we're going to let y and c be linear functions of parameter vectors, u and v. Those are the circles that on the previous slide, or the data points on the previous slide. And then our optimization problem is we need to find u and v that minimize the sort of least squares discrepancy between our data and our model. And this can be written as a Frobenius norm, which is weighted by this sigma matrix, which is basically just 1 over the experimental errors. So, okay, so the problem is that this optimization problem is ill-posed because we have way more parameters than the data can actually restrain. And it's also nonlinear, meaning that you can't just write down a really quick solution. So our strategy for this is for addressing ill-posedness, we're going to use regularization, excuse me. And to solve the nonlinear optimization, we're going to use an algorithm called alternating least squares. And now you can see where the name REGALS comes from. It's regularization with alternating least squares. Here's what that algorithm actually looks like. So in ALS, we're going to solve two linear least squares problems sequentially and then iterate. And regularization is brought in at each step. So first, we need an initial guess for one of the sets of parameters. Then we fit the concentration basis vectors by solving this linear least squares problem. And then we fit the SAXS profiles by solving a different least squares problem. And then it's necessary to apply some restraint, so to actually denormalize one basis vector or the other, and then we just iterate. Now, there's some other terms here that we're minimizing in addition to chi-squared. These are the regularization terms, and these are included in order to enforce smoothness in either time or Q or R space. And these functions B are approximating the squared magnitude of the second derivative of the basis vector. So this, by minimizing B, you make the basis vector smooth. And then each component has a regularization parameter lambda associated with it. And the magnitude of lambda determines how smooth that thing wants to be. Okay. So let's do some examples. As a first example, this is the AEX-SAXS dataset that I showed earlier. The issue with AEX-SAXS is that we have a salt gradient that produces a changing background. So the protein is charged and it binds to the column, and then you elute the protein from the column by increasing the salt concentration in a linear fashion. It's a little bit hard to see the consequences of that in this three-dimensional figure, but if you now integrate along Q and plot this is sort of the total integrated intensity versus frame number, you can see that characteristic rise, and that's the scattering from the NaCl in the buffer. Okay, and as I said, if you do SVD on this thing, you see 4 significant singular values. And one kind of strange thing is if you just do the SVD of this rising buffer part, there are actually 2 significant singular values, and that suggests that you need at least 2 components to describe the background scattering. Okay, so the next step is to define a model. And the best way to do that, I think, is to kind of make a sketch of what you think is happening. So here's my sketch. So we have a couple of background components that are probably very smooth and rising, and then a couple of protein peaks. And the way we define the model is we make these concentration functions of the smooth type, so linear interpolation of control points. And then for the SAXS profiles, we'll just leave them alone. So we'll just use the simple type. And then to make these background components smooth, we'll choose lambda very large, and then we won't do any smoothing on the protein components. Okay, so now what do we see? So this video on the right is showing the algorithm in more or less real time as we do 50 iterations of ALS. And it all happens mostly toward the beginning, so pretty quickly, so I'll rewind the video. So we start out from kind of sort of not a very good guess for what the basis vectors are. And then as we advance a few frames, you can start to see these concentration peaks now more resemble sort of what you expect to see. This is the reduced chi-squared versus frame number. This now approaches 1, signifying that we have a good fit to the data. And the SAXS profiles we get look pretty physical. And actually, I'm not showing this here, but we were able to actually fit crystal structures to these scattering profiles and get some insight into the enzyme that we were studying in this case. You can read more about that in this paper. Okay, so as a second example, this is ligand titration. This is kind of an interesting example because we had really, really nice data. So this is showing a titration of phenylalanine into a solution containing the enzyme phenylalanine hydroxylase, which is this tetramer that looks like a butterfly. And it was known that phenylalanine hydroxylase does a conformational change upon binding phenylalanine, which is its allosteric ligand. But we didn't know exactly what that change was. And so from 0 to 1 millimolar phenylalanine, which is kind of the physiological range of phenylalanine in your liver, you can see a change in the scattering profile at mid-Q, and this signifies a conformational change. But then as we went to higher phenylalanine, we start to see this increase at low Q, and that signifies an increase in the average molecular weight. So there's some high mass species starting to appear. And we'd actually done SEC-SAXS on this system as well, and so we knew that between 0 and 1 millimolar phenylalanine, this was not a completely pure solution. There was actually still a bit of this high mass thing present. So the sketch of what we have is like this. We have our resting state tetramer being the sort of dominant component at low phenylalanine. And then as we add more phenylalanine, we have an active tetramer that starts to be dominant. But all along, we have some uglymer, some large aggregate thing that we don't really know about. And it's at a low level at the beginning, and then it goes up at the end. So the way we sort of set up this model is to impose some boundary conditions. So here I'm going to force the concentration of the active tetramer to be 0 at 0 phenylalanine, which we knew was roughly true. And then we'll force the concentration of this resting tetramer to go to 0 at high phenylalanine, so above the physiological sort of range where this enzyme is fully active. And then we won't restrain this uglymer at all. Now, the way we're going to deal with the fact that these are different molecular weights is by imposing different limits on the maximum dimension. So we knew from the crystal structure that, and other methods, that the maximum dimension of this tetramer is around 130 angstroms, and it's basically the same in active conformation. So we'll set 130 angstroms for both. And then the ugly mer will just set a very large number. In this case, 300 angstroms is roughly the largest object you could measure on our beamline, given the configuration of the detector and so on. So here's the result we get. So it's actually pretty amazing. We are able to recover concentration profiles that look quite a bit like the sketch. It fits the data fairly well, except maybe at the highest phenylalanine concentrations. And also, we, for free, get these P of R functions for each component. And these can be analyzed further using sort of model building methods, which again, I won't talk about, but you can read about in our paper in JAX. Okay, so this also works on time resolved data, and this was fun. So we hadn't taken any time resolved data in the lab, so we looked on open sort of databases to see who had deposited interesting data sets, and these are a couple that we found. So one is a rapid stop flow mixing data set on this protein MSBA, actually the nucleotide binding domains of that protein being mixed with ATP. And what happens is the protein is initially monomeric, and then it becomes transiently dimeric and then falls apart again. And just with some very simple restraints, we're able to recover scattering profiles of monomer and dimer that agree quite well with structures derived from crystallography. We also analyzed this very interesting laser T-jump experiment. So this is done on a protein CypA, which was rapidly heated with a laser over tens of nanoseconds by about 10 degrees Celsius, and then they collected SAXS wax curves over a large time delay. And what's plotted here are difference profiles, so the laser on minus laser off. And weirdly, the scattering from difference profiles happens to be negative in this case. But we are able to analyze this data set using 3 components. And the physical meaning of these components isn't entirely clear, but one thing that it did allow is that we were able to reconstruct the sort of time evolution of the P of R function. And from that, we can get some more intuition. So this is now the laser on minus laser off, late time points minus early time points P of R function. And at early time delays, you have an increase in scattering around the maximum dimension of CypA, which is around 30 or 40 angstroms. And then that goes away, and then you have a decrease in scattering at longer length scales, suggesting that there's a change in protein-protein interactions. Okay, so how can you use REGALS? It's freely available for you to use. So we've put our code on GitHub. To further make this easy for people, we actually have 2 different implementations, or 2 equivalent implementations in different programming languages. And this was kind of our pandemic project. The student, Darren Shu, took my sort of original MATLAB code and translated it into Python and also made some improvements. And all of the examples that I showed in this presentation are included on this GitHub as standalone notebooks. So you can go in and analyze the data yourself. And in MATLAB, these are live notebooks, and in Python, they're Jupyter notebooks. Also of interest to SBGrid users, there is a graphical user interface for REGALS, which is going to be available in SBGrid soon. So this is a project by Jesse Hopkins at BioCat, who is one of the people who maintains this software package called BioXTAS Raw. This is an open source SAXS data analysis software. It's currently on SBGrid. And this is a sort of preview of the new REGALS graphical user interface. It's in the development version. So if you want to try it, you can install raw from source, which is actually not terribly difficult, or you can just wait for the next release on SBGrid. And I think this will be included in that next release. Okay. So that was part 1. Part 2 is we're going to look at scattering from crystals. Okay, so this was the picture I showed before, the scattering pattern of crystal with disorder. This is actually staph nuclease crystal collected actually in the mid 90s on a CCD detector by Mike Wall and in the lab of Saul Gruner. And this pattern is got a lot of interesting stuff in it. So, okay, there's Bragg peaks. This is what we typically would use to solve the structure of a protein. And the Bragg peaks tell you about the average structure. So we're trying to get the sort of mean electron density. This is what we would use. In this picture, you also see this cloudy diffuse scattering. That diffuse scattering comes about because of deviations from the average electron density. And the sort of holy grail of this field has been to try and use that diffuse scattering to learn something about protein dynamics. One great thing about diffuse scattering is that it's easy to calculate. So the total intensity, diffuse intensity, is just mathematically is just this. So it's the difference between the squared structure factor and the average. Sorry, the average of the squared structure factor and the squared average. So this F is not the structure factor of the unit cell, but it's the structure factor of the entire crystal. So structure factor is just the Fourier transform of electron density. And that's kind of important, and sometimes it's overlooked. Okay, but so here's what the equation says. Let's do some examples. Okay. Now, one thing to appreciate is that if there's no atomic motion, there's no diffuse scattering, and that's because the average of the square is equal to the square of the average. This becomes 0. So no atomic motion of this alpha helix, then there's no diffuse scattering. Now let's look at some other examples of different types of correlated motion. So if we have atoms that are moving independently, which is kind of represented with these spheres here, what you see is an isotropic, roughly, scattering pattern that's relatively featureless. Now let's look at a case where atoms are moving together as a rigid body. So that means this alpha helix is sort of vibrating up and down coherently. Now you see a distinct fingerprint in the diffuse scattering of that correlated motion. Now what's cool is these two examples, the way I've set them up, they have identical B factors. So these would give rise to identical Bragg diffraction and the same crystal structure. And you cannot distinguish between these two cases with Bragg diffraction. But with diffuse scattering, you can. Okay, so I showed atoms moving together as a rigid body. Now, what happens if the motions of those atoms are correlated with those of neighboring unit cells? Now, to give an example of that, what I did is I made this sort of simple lattice model where each alpha helix here is packed into this crystalline array, and I just put springs between the atoms in neighboring unit cells, and I let this thing vibrate at room temperature. Here's the pattern you see. So now we have sort of features that are almost kind of like Bragg peaks. They're actually at the Bragg peak locations, but they're much broader. And we refer to these features as halos. Okay, so again, so if the unit cells are completely uncorrelated, you can get this sort of smooth pattern. But if the correlations of the rigid body motion are across the unit cell boundary, then you see halos. All right, so actually what is known about protein diffuse scattering? It's actually been studied for quite a while, although you can put all of the diffuse scattering patterns that have ever been collected on one slide, and that's what's shown here. So this is basically everything that's been done since about 1980. And one thing you'll notice is that the sort of quality of the data increases, and that's because we've gotten better detectors over time. And in particular, more recently, we're able to collect three-dimensional maps of diffuse scattering by rotating the crystal during data collection, as you would do with Bragg diffraction. And okay, but there's an issue, and that is that data quality has still really made interpretation difficult. So because these detectors are fairly new, we've only had sort of quantitative model data comparisons for a short period of time. And in those cases that we have, the images were processed heavily to remove intense Bragg peaks. And as a result, you only have kind of a coarse map of diffuse scattering. Many models of internal motion have been tried, and most of these show really just kind of a modest correlation with the experiment. So correlation coefficients maybe of 0.6. So like here's kind of an example where we have, this is a slice through the 3D map of diffuse scattering from this protein CYP-A, and it's got some cloudy features. And then this is what a map that's calculated using a normal modes model. And if you sort of stare at this, you can see that it's, you know, some of these features are observed in both things, but it's not really a perfect match. So it's hard to know whether this normal modes model is appropriate or not. And then on top of that, others have argued that this internal motion is actually not the dominant contribution to diffuse scattering. It's actually lattice dynamics. And so the problem has been, though, that these maps that we have, which are quantitative, are missing the near Bragg data. So we don't really know the role of long-range correlations in these crystals. Okay, so when we set out to do diffuse scattering, one thing we wanted to check is how accurately we can measure diffuse scattering near the Bragg peaks. And it turns out you can actually measure this quite accurately using modern detectors. So this is a pixel array detector, the Lattice 6M, that we had access to at Chess when we started. And here's just a single sort of zoom in of a diffraction image from lysozyme. This is the tetragonal form at room temperature. You zoom in on one of these Bragg peaks. Now, these are the actual sort of pixel values of intensity on the detector on a logarithmic scale. What you see immediately is that most of the Bragg intensity is in like 2 or 3 pixels, which is pretty amazing. And it decays very rapidly to this background level. So we're able to measure 10 to the 5th photons in a pixel right next to a pixel with like just a few photons recorded. So we're actually able to measure this background intensity fairly close to the Bragg peak. And that's because pixel array detectors have this great property of having high dynamic range and also a point spread function that's just around the size of 1 pixel. Okay, so we've got good detectors now. Let's make detailed maps of diffuse scattering. The system we chose to study first is lysozyme in the triclinic space group, P1. This is an ideal system to study diffuse scattering because, first of all, it's pretty simple. There's no symmetry averaging from space group symmetry. You just have one protein per unit cell. These crystals also have low mosaicity and they diffract to high resolution at room temperature. And because of the small unit cell, the Bragg peaks are going to be well spaced on the detector, so we'll get a lot of samples between the Bragg peaks. And what we decided to do was data collection at room temperature using this sort of standard plastic capillary system. And then we include as well background measurements. So this is scattering from the capillary itself and also the air in the beam path. And then we just do a simple subtraction. So crystal diffraction minus background. Okay. Now to merge all these images together, we needed to make a new scaling method. And this is what we came up with. So first you have to correct for geometric effects. So things like solid angle, polarization, pixel efficiency on the detector. This is sort of normally what you would do for Bragg data as well. And then the second step is we need to globally refine a scaling model. And so this is to bring all the individual images onto the same intensity scale so we can merge them together. And we found that it was necessary to have to include a lot of different effects in the scaling model. So one of them is we have an absorption term. So this is sort of small few percent corrections across the face of the detector that may change with spindle angle. We found that we had to also correct for different gain values of the different chips on the detector. And then of course we need a per image scale factor because the path length through the crystal changes as you rotate. And then finally, we needed an isotropic scattering offset correction. And what this is correcting for bits of extra solvent or mounting material that pass through the beam as you rotate. Okay, so after all of that, then we also developed a method to place the intensity on an absolute scale of electron units. And this removes some of the arbitrariness of model fitting, because the model has to also predict the intensity of the diffuse scattering. And I won't say anything more about this absolute scale thing, but you can read about it in our paper. Okay. So to get a complete data set with high redundancy and good signal to noise, we collected 11 wedges of 50 degrees from 4 different crystals. The crystals, as you can see, are fairly large. The scale bar is 300 microns. And we needed large crystals because crystals damage quickly at room temperature compared with cryo. So that was needed to get good signal to noise. Okay, and then we refined the scaling model for all of these guys, merge in 3D, and we get a 3-dimensional map of diffuse scattering that we affectionately refer to as the Death Star. This map has a resolution of 1.25 angstroms. There are 1,573 voxels around every Bragg peak, so it's quite a detailed map of diffuse scattering, and that results in a fairly large data set of about 50 million voxels in the sort of upper half plane, or upper half hemisphere, I guess. Okay, so to kind of appreciate this, I'm going to make a slice, show you a movie that slices through this map in 3D. And so here we're moving a plane through the 3-dimensional volume of the diffuse scattering. And what you can see immediately is that every once in a while, you see a bunch of bright red spots. And these happen whenever we are at a lattice plane. So whenever L here is an integer, and they coincide with the Bragg peak locations. And these are the halos that I alluded to earlier. Now, in between, so for example, if we go to a half integer plane, you don't see halos, but you do see this very interesting cloudy pattern. And this is coming about from short range correlations in the crystal. Okay. Now, the first thing we do is we needed to understand sort of the origin of these halos. And so what we did is we just developed a very simple vibrational model where we have rigid body motions of each protein in the lattice. And again, I connected the protein to its neighbors using springs and let the thing vibrate at room temperature. And then what we did is we refined these spring constants in order to reproduce or best fit about 1% of the diffuse peaks. And in this particular slice, those peaks we used for refinement are surrounded by blue boxes. So here now is the simulated pattern after refining this model with a zoom in here, and this is the pattern we measure. And what you can see immediately is that we're able to reproduce, the intensity of these halos, the sort of odd shapes that they have, which is quite amazing considering that we only used a few of them to do the refinement. And that's partly expected because there aren't a lot of degrees of freedom in this in sort of lattice vibration models. So hopefully a simple model can explain the whole thing, and that was the case here. Now, what we didn't expect was that the model also predicted a lot of cloudy diffuse scattering between the Bragg peaks. So I said earlier that this cloudy pattern is related to short range correlations. And so people had naively assumed that would be dominated by internal motions of the protein. But here we're saying, no, these rigid body motions also contribute a cloudy pattern. And actually, it's pretty similar to the cloudy pattern we see in the measured data. Okay, so that was a little worrying. Is rigid body motion responsible for all of the diffuse scattering that we see? So to understand this better, we looked at the data in a different way, and that is we computed the diffuse Patterson function, which is the Fourier transform of the diffuse intensity. Sometimes this is called the 3D delta PDF. And it's equal to the mean autocorrelation of the electron density difference, meaning the instantaneous electron density of the crystal minus the average value. And what you can think of this, so here's a slice through this Patterson map. And you can think of a vector in this map as being a vector from the origin as being a vector between two atoms in the crystal. So if we zoom in on the origin, these are going to be the pair distances in the crystal that are small, which we would hope would be, mostly within a protein. And so here I've just drawn a circle showing you what 10 angstroms looks like, which is, this is going to be like roughly the size of the lysozyme molecule. And you can see these kind of intense ripples in the, or features in this Patterson function. Okay, so how much of this is coming from internal motion and how much is from lattice dynamics? So what we did is we just, again, simulated the diffuse scattering from our lattice model that we'd refined and calculate the Patterson function, and then compare that with the one we measure. What you can see immediately is that the measured one has more intense features at low, short distances than does the lattice model. And so we conclude that the lattice dynamics alone really can't explain these correlations at short range. Okay, so now let's look at the B factors. So here is in sort of shaded in gray, these are the total B factors for lysozyme averaged over every residue. And they vary quite a bit across the protein. Now, if we just calculate from the lattice model what the B factors ought to be, that's this blue line. And you can see there's a difference. So this difference must be due to internal motion of the atoms. Okay, so how do we model that? What we did is we made a normal modes model of using a crystalline elastic network, so it's just sort of an all-atom model with springs that vibrates. And then we, again, refine the spring constants, but this time we fit this B factor difference, the difference between lattice and internal motion, using the model. And that's what these crosses, these symbols show, is the B factors after fitting. And they actually are able to reproduce a lot of the variation in the PDB file. Okay, now we simulate, again, diffuse scattering, calculate Patterson function. So these are the ones I showed already, lattice alone measured. On the right now is the Patterson function from that internal motions model. And you can see that this internal motions alone doesn't look really much like the measured diffuse scattering, but we're going to add it to the lattice model because both of those things are contributing to the diffuse scattering. So adding those two together, now we get this map. And I think I could convince you that this is now starting to look a whole lot like what we measure. So we can be quantitative about this too, but I think just looking at the maps actually is pretty good. So you can say, okay, once we've added in this internal motion, now we're getting pretty close to the actual measurement. So as I've said, we can be quantitative about this too. This is now calculating a correlation coefficient as a function of resolution shells. This is kind of like a FSC or something for our Patterson function. And blue here is showing the lattice model alone. Red is the lattice plus internal, so we get a boost in CC basically at all resolution shells. So using the CC, we can evaluate models quantitatively, and actually a big part of this project was doing all atom molecular dynamics, which was done in collaboration with David Case. And one thing that he did is he did all atom MD on different unit cell sizes or different supercell sizes. So in other words, he arranged proteins into a crystal in the computer with periodic boundary conditions and then ran molecular dynamics. If there's just one chain, you get a pattern like that. And as you increase to more and more and more, eventually reaching 343 lysozyme molecules in this simulated crystal, you start to reproduce diffuse scattering patterns that look an awful lot like the measured ones. But using this quantitative measure, actually the MD is not doing as well as this lattice dynamics model. That's not because MD is particularly wrong. It's just that the lattice model has the benefit that we know the position of every single atom from the PDB file. MD is completely unrestrained, so its initial condition comes from the PDB file. but it's free to move. And it turns out that in this MD simulation, the structure tends to drift a little bit from the crystal structure. And we believe that's actually mainly what's responsible for this sort of less ideal agreement. Okay, so finally, what about correlations of biochemical interest? So in lysozyme, it's sort of well known that there's this hinge bending model for the motion of lysozyme. And basically the molecule can be thought of as having two sort of domains with a cleft, and the cleft is where the substrate binds. And the hinge bending mode of lysozyme is thought to be a kind of vibration of these two domains such that the cleft opens and closes. So does diffuse scattering support hinge bending? This is a hard question to answer. So our strategy was to just compare two different models. So we made one model shown on the left, which is the one I've shown you already, the elastic network model where we have sort of full dynamics. And then we made another model where we artificially damped out all of the modes associated with hinge bending. And that's shown in this plot of correlations between different residues that there's basically no correlations between beta and alpha now because we've damped out all those modes. But it turns out both of these models were refined to the B factors and they fit the B factors equally well. So which model agrees best with diffuse scattering? Now I showed this plot of CC before. Already the difference between the lattice model and lattice plus internal is pretty small. So we're going to have to zoom in. So now we just look at the difference, the sort of gain in CC above the lattice model. So red now is the full internal dynamics model, and now dashed line is showing the CC for the model where we've artificially suppressed these hinge bending modes. Yeah, and what you can see is the full dynamics model does give a better fit to the diffuse scattering. The improvement, though, is very modest, so it's just a 0.01 or less CC. It's very small improvement, but it is consistent across all resolutions, so that's a good sign. And I think this, we've got a long way to go before we get to perfect CC. So I think that maybe more realistic models, such as things like restrained MD simulations, might improve the fit even further. Okay, so that's kind of it for results, but I just want to point you to the fact that our diffuse scattering data and all the software are freely available on the internet. So software is on GitHub. It's a library written in MATLAB. The raw diffraction images have been deposited on the SBGrid databank, and that's shown here. And our 3D diffuse maps have also been deposited on the CXI-DB, so you can download those. And I just want to give a couple final thoughts before ending. So as I said, protein function, this is a hard problem to solve. It's going to be hard to debug the wiring diagram of proteins, and this is going to require new structural techniques that are sensitive to fluctuations. Diffuse scattering has a lot of potential as a tool to use for this. It reveals the dynamic origin of B factors, as I've shown. There was this question early on of, is it lattice dynamics? Is it internal motion? Diffuse scattering says, yes, it's both. Diffuse scattering you get for free when you measure a diffraction pattern, but you do have to measure it well. And then I just want to point out that, there's 176,000 structures in the PDB. The number of those structures that's associated with a well-measured, highly detailed diffuse map is of order 1. We need to get some more examples. So let's measure more diffuse scattering. And that's it. I'm happy to take questions.
Fantastic. Thank you very much, Steve. Questions, you can raise your hand. There's a reactions button, and there is a raise hand option there. That will bump you up to the top of the participants list, and I'll see your question. You can also send it by chat, and we will either call on you or bump you up. I know I've got a question. I think it comes right down to that last point of yours, some more samples. So like, things that come to mind, for me, different lattices, myoglobin, it's got an iron in it, right? So,
Yeah.
what sort of samples are you thinking about? Like this is my PhD advisor used to refer to these things as, the lighthouses, these derivatives, heavy atoms, or maybe that's just too complex. I don't know.
No, that's a great, okay, so I should say when we first started this diffuse scattering project, we were very ambitious and we thought we were going to do anomalous diffuse scattering. And we actually started on that methyltransferase enzyme I showed at the beginning, which has cobalt and it has iron. And that was hard. But the diffuse scattering pattern was there and it was very interesting looking. We just couldn't figure out what it meant. And that's why we went to the simpler system. So yeah, so now that I think we've cracked the simpler system, it's time to look at more complicated things. Crystal symmetry is going to be a complication. And we've actually already collected data from lysozyme and a number of other space groups that shows diffuse scattering and halos in the same way that P1 does. And I think we'd really like to have some examples that are of sort of genuine interest to biochemists. So, I think we all have a soft spot for lysozyme, but it's not the most exciting enzyme in 2021. So, like, I think we want to look at some more interesting ones too.
Yeah,
so that's kind of the direction I think we should go.
Yeah, I think you need, you probably are already, but just sort of lurking around, talking to the beamline scientists about like, what have you seen that's interesting, right? Because
yes,
they see these images coming in, right, and they know like, oh yeah, I saw one last week, it looked pretty good or something, right? But this is the shame that we don't. A lot of labs, their images are sitting in closets on tape, and it's kind of hard to get your hands on these things. So we had a couple other questions. I know Pete had some. So Pete, go ahead.
So I've two questions on both different areas. So I guess the first one for your diffuse scattering part, and that was amazingly cool, by the way, but that's not a question. The question that I was wondering about is there've been periodic discussions in X-ray crystallography about the distinction between the merging R factors for data collection and the R factors for refinement, and why is there this big gap that we can't figure out? I'm curious if you guys took a look with that. Does that gap go away in your P1 lysozyme system when you account for the diffuse scattering?
That's a great question. And actually that's a topic that David Case has been very interested in. He's collaborating with James Holton to try and address that problem. And this is the R factor gap issue.
Yep.
So it's thought that much of that is due to the failure to account for, on the one hand, protein dynamics in crystal structures, or the extent of true disorder, and also the solvent model, which in crystallography is a bit simple. It's usually just kind of a flat mask.
Yeah. So
I think the idea there is that, you could use MD to perhaps close that gap. And one thing that's great about this lysozyme system, the P1 lysozyme system, is the refinement actually was able to get R factors down to, I think, close to 10%, so pretty low for a crystal structure. That's partly because it was very high resolution. So that may be a good system to look into this R factor gap problem more closely.
Yeah, that's cool. I'll have to keep an eye out to see if they have anything coming out there, because based on what I've seen, the bulk solvent models for crystallography, they help, but they don't get rid of the R factor gap, and same for everything else that everybody's tried. So it's always exciting to see people trying to work on that.
Yeah.
I guess for second question, this may be an ill-posed question because of lack of linear algebra knowledge on my part, but when you were describing your REGALS algorithm, I was very much reminded of genetic programming approaches where you have a family of models versus just a single one. Is that an illusionary similarity, or is that something that's worth investigating, or something you've already investigated and is not worth the trouble?
So I think that there are, for difficult nonlinear optimization problems, there are sort of a lot of different tools that you could throw at it, genetic algorithms being one. And genetic algorithms are used quite a bit in small angle scattering data analysis, but we didn't use that for our system. What we used was actually, the alternating least squares has the advantage that it's deterministic, so there are no random moves. And so if you set it up, it may not be the fastest algorithm to converge, but it should give you the same answer every time. And we felt like that was a good feature.
Yeah, but when you're talking about families of potential models and nonlinear, or non-convex global optimization, it's always something that pops up. So great. Well, thank you. And once again, thanks. That was a very interesting talk.
Yeah, it's really fantastic. Any other questions? We're a little long, so we can go ahead and wrap up and sort of formally wrap up the talk. But if others want to stick around and discuss, I don't know if Steve's got time, but we could. And
happy to chat.
Yeah, it's one of the benefits of the sort of online platform. But yeah, thank you very much.